In each of your cells right now, billions of reactions are taking place to keep you alive. For that, you can thank enzymes, tiny molecular machines which form long assembly lines that turn non-living matter into the building blocks of life. In fact, what we call life is a single continuous chemical reaction that started 4 billion years ago and has lasted in an unbroken chain leading all the way to you and every other living thing on this planet. So what separates life from a regular chemical reaction? The answer is our genes. When you think of genes, you probably imagine DNA, a molecule that acts as a blueprint, storing the information required to build a living organism. You've probably heard that genes determine your height or the color of your eyes. But when you look closely, there's no single gene that controls these traits. In fact, there are 16 genes that influence eye color alone. So, if one gene can't even control eye color, then how do genes build an entire eye, let alone a whole human being? To understand, we've got to go back in time, even before we realized that genes were made of DNA.
The year is 1948 and the theories of Charles Darwin and Gregor Mendel have been combined in the modern synthesis. We know from Mendel that certain factors are passed down from parent to child and control specific traits of an organism. We call these heritable factors genes. From Darwin, we have learned that mutations or changes in these genes can accumulate over time, leading to evolution. Despite all this, one question remains a mystery. How does a gene result in a visible trait? The answer lies in mutants. However, mutants are far too rare in the wild. So in order to study mutants, we must create them.
But in order to do that, scientists needed a model organism. Fruit flies? Too complex. E. coli? Too asexual. But what's this? Enter Neurospora, whose name translates to nerve spore, also known as the red mold. This versatile fungus only needs 3 things to survive: sugar, salts, and biotin. This is called a minimal medium. With just these 3 things, Neurospora can create everything else it needs using its enzymes. To reproduce, it produces a structure known as an ascus containing spores. These spores are haploid, and so they don't have a second backup copy of their genes. That's why, by bombarding these spores with X-rays, we can create mutations. However, unlike in the comics, most mutations are harmful, and some are deadly. These cells are now damaged, and many cannot produce what they need to survive from the minimal medium. However, these mutants might stand a chance with complete medium. This is a mixture containing everything a fungus needs to survive, meaning that it doesn't need to make anything for itself. A fungus that dies on minimal media, but can survive on complex media, means that it has lost the ability to produce an essential nutrient. By taking spores from these mutants and cloning them, you can pinpoint what has changed. Each of these tubes contains minimal medium with only a single vitamin or amino acid. Again, most spores will die. But look, a survivor. This mutant has lost the ability to make arginine, and now it must scavenge it from its environment. Because this tube contains arginine, it's able to live. For rare mutants such as these, we can infer that one of the enzymes used to make this molecule has suddenly been broken. Changes in the genes caused by X-rays have therefore changed the functionality of one of the cell's molecular machines. In other words, one gene stores the information for one enzyme. However, we've come a long way since then. We now know that genes don't just code for enzymes, but for sequences of RNA. These lets us build any protein in the body, not just enzymes. Some RNA sequences can also inhibit others, giving genes a high level of control over the cell. Genes then use these abilities to build the environment around them. They also cooperate with other genes, making more and more complex structures. And they do all of this to make more copies of themselves. From a gene's perspective, you are simply an automated chemical meat suit that continues the gene's survival. But by understanding how genes work, we can take back the wheel. We can cure diseases, live longer, and continue this one big chemical reaction that we call life.